Good morning, everybody. Where are we at today? Get out the chat. All right, let's see. Where were we on Friday? This is pretty much where we're at. So we finished up with RNA pol. I think I might have said that was in a prokaryote. I'm wrong. This is a eukaryotic RNA polymerase because only eukaryotes have 3 different types of RNA polymerase. Bacteria only have 1. So I think I said that wrong in the video. I was just checking back, see where we're at. Okay, so basically there's 3 kind of general steps to transcription. All right, and these are shared between prokaryotes and eukaryotes, but some of the details differ, particularly in the initiation and the termination of transcription. So in bacteria, and we'll start with bacteria first, we've got essentially 2 main consensus sequences, right? And so as we talked about last Friday, we have essentially 2 parts to any given gene, and this applies to bacteria and to eukaryotes. We have the start of transcription, so the bit of the gene that's essentially contains the information, right, required for that gene product to do its job, whatever that might be. And we also have upstream of that, so kind of before, that's what we mean by upstream. So before the start of transcription or the transcription start site, we have what's called a promoter. And so promoters are, it's a little bit different in bacteria because they have operators as well, which is kind of part of the promoter. But essentially they're the region that of DNA that, come on, up, bring it up. What do you want to do? Puppy being curious. Oh, nice job, Luna. Hey, get out of there. Not the most appealing view of my dog at 9 o'clock on Monday morning. They're the region to which RNA polymerase binds, right? So that's the main thing in many ways, but also a bunch of other proteins collectively called transcription factors, right? And as well, things which might turn off transcription. So we're going to get into that in huge detail in the 4th section of the course, right? The control of gene expression, because there's a lot to it. But for now, we're just going to go on, breakfast. Go on. We're going to talk about just in terms of RNA polymerase binding and the consensus sequences there. And so what we mean by consensus sequence, we'll come back to this in a second, is this is the sequence that you find most commonly at that position. And so this is like a kind of a pictorial way of showing that, right? So in certain positions, you'll have only an A or a G. Other positions, you'll mostly have one nucleotide and sometimes others, right? And so essentially what this is if you look at either in one species, a bunch of different genes, or between species, you'll find the same bases quite commonly in that position, right? Whereas other regions of DNA have no particular commonality, right? So it's like it's kind of like the average, right? What you would find on average. Doesn't mean that that's always the sequence, right? Because for example, this is the Tata box. Well, the Tata sequence, Tata boxes in eukaryotes. And you can see this is the consensus sequence, TAT, AAT, right? Again, remember, low bond strength between those bases, right? So this is a region of the promoter which is going to get opened up by RNA polymerase before the start of transcription. Right, so again, that kind of common theme that we've been talking about. And so down here, Rec-A, which is involved in recombination and DNA repair from memory, that has the exact same sequence as the consensus sequence. Others, like LAC-I, which is in lac operon inhibitor, don't hew really as closely to it at all. So these sequences in bacteria are always in the same position relative to the start of transcription, right? So if the start is +1, you'll always find the sequence beginning at basically 10 bases upstream of that. And then there's another one at 35 bases upstream, roughly speaking. Obviously, there's differences between them here. These aren't kind of missing bits of DNA. These are simply gaps introduced to make that alignment work, right? So that all those bases line up properly. Another point as well is that how closely these consensus sequences in individual promoters match the consensus determines how strongly RNA polymerase binds. Right, so essentially, you have kind of strong and weak promoters, right? And what we mean by that is that that reflects how efficiently RNA polymerase binds, or really like the whole caboodle, right? Because there's a lot of extra. What do you want? Oh, here's the other one. As you can see, she's been getting a lot bigger, haven't she? So yeah, there's a lot of different proteins involved, but generally speaking, strong promoters are more likely to bind that apparatus and result in transcription of that gene. Right, so when we talk about strong and weak promoters, what we're really talking about is how frequently does that gene get transcribed? How likely is it that you're going to get a gene product from it? And so really, in a way, it kind of reflects how important or how much of that gene product is needed. So if we go back up here, so for example, hello girls, LAC-I is an inhibitor of the lac operon, and it's produced all the time, whether or not lactose is present or not, right? But it's produced at a relatively low level because sorry, a little bit distracted. Hey, love, bit distracted. No. Yes, where was I? Okay, so LAC-I is always produced, but it's produced at a low level. And if you look at the consensus sequences, they're pretty far from the average, right? The consensus. Whereas LAC, the actual lac operon product, right, the one that produces the stuff that turns lactose into glucose, has a much closer match to the consensus sequence, right? And so when you need to turn lactose into glucose, you need to do, a lot of it basically, right? Because you don't have any glucose around. And so you can actually look just at these two genes and see, kind of get an insight into, that strong versus weak promoter deal. All right, so we really have to think of it in the context of what does that gene product do, right? And how do you need it, essentially? So also, obviously, mutations in these regions can affect that promoter strength, right? So in many ways, the reason why LAC-I has like a divergent consensus sequence is because it doesn't actually matter, right? So if there's a mutation in the consensus sequence for LAC-I, you don't need a lot of it, right? So there's no selective pressure to weed out those mutations. And so that's how it diverges, essentially. So, hey, get off of that. In bacteria, it's relatively straightforward, right? So kind of the big difference between bacteria and eukaryotes in terms of transcription is that transcription initiation in bacteria is controlled really by the presence or absence of repressor proteins, right? And so bacterial genes by default are always on, right? So they're always available to be turned on unless they're, turned off, essentially, right? Whereas eukaryotes are very different. Those are by default off and they have to be turned on. So the turning on part is a lot more complicated in eukaryotes. So in bacteria, all we really need is a protein called the sigma factor, right? So the sigma factor is a transcription factor. Let's type this out. And basically its role is to guide the RNA polymerase to the promoter. Right, so it's pretty straightforward. Essentially what happens then is that, all of the proteins required for the polymerase to do its job, which there are many, right, assemble on the promoter, kind of glom on. And, oh, that reminds me, I need to upload this PowerPoint because we've got a video on slide 15. And essentially the RNA polymerase kind of pops open the DNA strand in the promoter using those weaker TA regions, and then simply synthesizes a strand of RNA, right? That's pretty much all there is to it in bacteria. Really quite simple, relatively speaking. So a simple matter of sigma factor binds, RNA polymerase binds to that and to the promoter, and you get transcription, right? So that's what I mean by transcription being always on, right? Unless there's something to physically stop this process, transcription will always occur from a gene in bacteria, right? It's like having the light switches in your house and every room are on, unless you go around like I do after my kids and turn them off. That's my karmic punishment for making my dad do that to me when I was a kid. Life's a bitch, I guess. Comes around and bites you on the ass. So anyway, it's actually kind of funny to see how much you turn into your parents as you grow older. That's probably a horrible thought for many of you that are on the younger side, but it's a funny one, particularly now I'm in that position. Anyway, so real simple, right? Now, eukaryotes are very complex, right? And that's, no sugarcoating it whatsoever. And one kind of reflection of that, where we've got the same transcription start site kind of deal. Now we have a lot less kind of consensus, right? So we have something called the Tata box, right? So the Tata box is kind of equivalent to the -10 sequence in bacteria, but it's also a recognition site for another protein called the Tata box binding protein. So the Tata box is both kind of a weak point in the promoter where the RNA pol opens the DNA, and also a recognition site for the box binding protein, TBP. And that recruits RNA pol to the promoter. So kind of in some ways, the TADA-box binding protein is similar to the sigma factor in bacteria in some ways. So what you can see though is that it's much smaller, and that's it in terms of commonality, right? So there's a lot less commonality between promoters and eukaryotes, and promoter sequence in eukaryotes can be huge, right? So in bacteria, they're pretty well defined and they're pretty small, right? So, you know, typically in the hundreds of base pairs. In eukaryotes, and just skipping ahead here, we have a similar promoter called the core promoter, right? So the core promoter equals RNA pol binding, assembly. Basically, that's kind of like the Cliff Notes version, right? Then, it's also called the minimal promoter, which basically means it's the smallest kind of bit of DNA that will allow transcription from of that gene, essentially, right? So it's the bare minimum that you need for this gene to be transcribed. Now that's the core promoter. That's, you know, again, in the 400 to 500 base pair range, give or take. Now the regulatory promoter, well, that can go for literally miles upstream, right? And it's very hard to define, right? These things are extraordinarily difficult to figure out, right? What is needed and what binds where, right? And so these regulatory promoters essentially what modify the rate of transcription from that promoter. And it's also undefined, right? Because it's really hard to figure out where it stops, right? Presumably at the next gene, but not always, right? And basically it controls how much in what cells, you know, tissues, when, in response to what. That gene gets expressed. So already you can see like, oh, this is just like way more crazy than bacteria. There's a lot more going on. Oh, bloody hell, they're doing the road again. It's the downside of having a white car. It just looks like crap whenever it gets dirty. One thing as well, which I often forget because, you know, I kind of know it. One thing that's really worth kind of just really pointing out, okay, and this is something that seems sort of obvious, but really bears repeating, right? There are proteins which bind DNA. Oh, what a bloody mess they're making. And what they do is they recognize a particular shape of the DNA and a sequence. Again, it seems kind of, you know, almost self-explanatory, right? You know, the DNA binding proteins, they bind DNA. But that requirement of a specific shape and sequence is really, really critical to any aspect of gene expression, transcription regulation, and so on, right? We have to have proteins that recognize specific regions of DNA because otherwise, for example, RNA polymerase would not know where to bind, right? So sigma factor comes in, binds, I think the -10 box, I'm not actually 100% sure, and recruits RNA polymerase, right? So it's kind of its ability to recognize that sequence is critical to its function, right? That's also why if you mutate or you have a mutation in that sequence, that protein may not recognize it anymore or may do so less well. Okay, so these sequences and the proteins that recognize them are, central to how this all works. And if anybody has any questions at any point, please do stop me. So in eukaryotes, we've got a whole bunch of transcription factors. These are the general transcription factors. I don't know what happened to G. Maybe that was, on holiday at the time. But we've got A or C as well. Huh. Anyway, and these ones are all involved with RNA polymerase binding, right? So again, this is a multi-step process involving multiple proteins all working together in particular ways to affect an outcome. And that's actually pretty common to an awful lot of biology, to be honest. Okay, so you ready? All right, let's blow this up a little bit so it's a bit bigger. Okay, so again, get back. In a particular sequence. Right. So first up, again, we have this core promoter and regulatory promoter. We'll get back to this in a second. So in that core promoter, you find the TADA box, this thing. And that's recognized by two proteins, the TADA box binding protein, which is kind of self-explanatory, and transcription factor 2D. Right, so these bind, they recognize that core promoter, they come in and they bind and they sit there. And by doing so, they kind of like wave a flag, like, come on, here I am, come bind to me. And so all of these other proteins now begin assembling on and around those two proteins, right? So those are the kind of the pioneers or scouts or whatever you want to call them that find the TADA box and then bring everything else in. Okay, so we all assemble all of these different proteins. We've got RNA polymerase 2, right, which is obviously the thing that's going to be doing the transcription. We have 5 other transcription factors that also recognize other DNA sequences in the core promoter. And we have a larger protein called the mediator. And so the mediator is really important for essentially mediating interactions between this whole complex and activator proteins, right? So this is the intermediate, essentially. Okay, so once we get to that point, transcription may start, right? It just doesn't do so very well. So we've got everything on the promoter, but unlike bacteria, it just sits there, right? So we've assembled the whole machinery, but we haven't started it yet. This is where that extra level of complexity kind of kicks in with eukaryotes. And it's also part of the whole gene expression has to be turned on in eukaryotes, right? By default, it is off. So we need something to activate this complex and get it going. And funnily enough, these are called transcriptional activator proteins. And so these may bind to regions in the regulatory promoter, right, that's next to the gene. It's called cis regulation because it's kind of, next to. Or they might bind regions in the DNA way, way farther away. And actually, fun fact, let's see if I'll open this. This was actually, just want to show you a fun picture that a student sent me. This Mr. Tuxedo, there you go, from a student in, I think it's summer or fall, spring, something like that. Can't remember. Anyway, there's a gene in us called Sonic Hedgehog. Don't ask me why. Supposedly, as the guy who discovered it, his son was playing that game a lot on the Sega. Anyway, this controls, in part, digit identity, right? And basically, you get a lot of it down here on this little limb bud. And that means this is pinky. Get a little bit less, that's whatever that one is. And I don't actually know what the different fingers are called. There's a ring finger in there somewhere. Oh, that's the ring finger. And essentially you get none, and that's why you have a thumb, right? And so that is controlled by an enhancer that's way, way far away, hundreds of thousands of bases away from the gene. And that binds a transcriptional activator protein, which controls transcription. So if you screw it up, you get cats with multiple extra toes. It's called polydactyly, which is just a super fun thing to think about. And anyway, when I was, see, Mr. Tuxedo doesn't look especially happy about this. But when I was talking about this, one of my students was like, ah, I've got a cat just like that. And so she sent me some pictures. And so you can see there's 1, 2, 3, 4, 5, 6, maybe 7. So yeah, I think the record is 36 toes on a cat, which you have to kind of think about like how that's divided up. It's obviously not all on one paw. But anyway, so that's a great example of that upstream enhancer region. All right, we'll go into that in more detail later. It'll make a bit more sense. So anyway, when that binds, and there's also proteins that kind of bend the DNA, right? So it's actually physically bends the DNA around. That brings that transcriptional activator protein into contact with the RNA polymerase, and all of this jazz all together, get back, activates RNA polymerase, and off it goes. And so if we go back to, actually, I think I might have it open in my browser still. Oh, sorry, getting a little bit distracted by looking at animated videos on YouTube. So essentially that's what we're looking at here, right? So this is in eukaryotes. And what you're seeing here is the Tata box binding protein, right? You can't actually tell where the gene is, right? Because it's just DNA, right? Tata box binding protein and TF2D is here. We're going to see a whole bunch of other proteins assemble, including the big ones, which make up RNA pol. And then we're going to see the DNA kind of curving around. So here, everything kind of assembles, forms the right shape. It doesn't actually make any noise. Right? I'm pretty sure that purple E sign thing is RNA pol. I could be wrong. So this is the complex we're talking about here. All of this stuff, right? And so obviously they're just made as like funky circles and stuff. They don't look like that. They actually look like this. And what you'll see now is that this DNA upstream, right? So the gene essentially goes down to the bottom left. That DNA is going to get bent round. See, nothing happens. And that is a transcription of that activated protein bound to an enhancer, right? And that's shown as being fairly close, right? So that might be in the regulatory promoter right next to the gene. Could also be, again, as I said, you know, completely different part of the chromosome. And so now the whole thing is activated and we're off at the races, right? That's pretty much it. And so that video is up there for you, should you kind of want to look into it in more detail. So really, in terms of the actual process of transcription, there's nothing very much to it. RNA polymerase does really all that we've said before, right? It goes along the DNA, opening up a little bit of it as it goes to create this single-stranded complex. RNA nucleotides come in, NTPs, and get added to the 3' end of the growing RNA strand based on base pair complementarity. Right, that's really all there is to it. So the actual transcription part of transcription is, you know, pardon me, fairly mundane. But we also have to stop it at some point, right? Because, you know, this isn't free. And so you don't want to be transcribing your entire chromosome or genome if you're a bacteria. So you have to stop this at some point. And so there are actually many different, well, there's at least 3 different ways of doing it, at least 2 in bacteria. And yeah, eukaryotes are kind of, as you'd expect, more complex and less well defined, right? Part of the reason is bacteria are much simpler to work on, right? It's kind of the easy way of looking at it. You know, they have a much smaller genome. They're kind of easier to grow and, you know, screw around with, essentially. And it's a lot easier to see problems actually visualizing stuff than in animals. That's actually why yeast is such a powerful model organism for research, because it's a eukaryote, but it's also single-celled. So it's kind of equivalent to a bacteria in a sense. So anyway, bacteria actually is a lot more kind of well-defined. And as far as we know so far, there are 2 main kind of means of doing so. And these actually, remember how I was, you know, waxing lyrical about RNA and all the many shapes it can form, how complex it can be? That is actually part of how this works, right? And so one thing that I really want to, you know, bang my drum about is this formation of what are called secondary structures in RNA, right? And this, what you see here, is what's called a hairpin loop. And it forms because it basically has 2 regions, there's a bit here and a bit here, they're inverted repeats. And so when you make an RNA, essentially you have 2 bits that match, right? And so when you transcribe that RNA, those 2 bits can then base pair, right, and form a loop, right? This is critical to a lot of, particularly in bacteria, a lot of different aspects of transcription, right? So kind of keep that in mind, right? So these RNA molecules can form structures, and those structures have a role. They're very important. And this actually, transcription termination is one of the aspects of this whole lot that students struggle with a little bit, right? Because there's just different concepts at play, essentially. And so we have 2 types of transcription termination. Row dependent, so it requires on a protein called Rho. Row, row your boat gently down the stream, merrily, 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 like such a dream. That's how I remember it. And intrinsic termination, right? Which basically relies on the actual sequence of the RNA being produced, the mRNA being produced to terminate its own transcription. Right. And so there's also another aspect, and this is also common to both, right? Is that RNA polymerases, actually DNA polymerases too, they don't like strings of the same letter. I don't know why, but it seems to kind of cause them to pause their, they're like, ooh, not too sure about this. Hang on, chaps. And they slow down, right? And so actually, that's also one of the things that causes mutation during DNA replication too, are these kind of long string of the same bases. And so in bacteria, you have, for intrinsic termination, you have two things going on. One, you have a region that slows the RNA polymerase down because it's happily just chuntering along, like in that video. Right? So you have to slow it down. And the second thing is the formation of what's called a terminator loop. Right? That's in intrinsic termination. So first you've got to slow down the RNA polymerase, right? So it doesn't just zip past before this terminator loop can form. And then the RNA polymerase itself has made the bit of DNA that's going to pop into a hairpin loop, which will stop it from doing more transcription. And so the sequence that stops transcription is built into the sequence of that gene, right? That's what we mean by intrinsic, right? It's part of the gene. Obviously not part of the protein coding bit, but it's part of the actual mRNA that's produced. And so essentially what happens is we have a string of A's, right? And we have these two inverted repeats, right? So if you remember, we come across these in transposons too. They're what transposase recognizes. However, in bacteria, at least in transcription, these inverted repeats are able to kind of flip together and base pair to each other, right? So this is a structure, a bit of double-stranded RNA, right? Which doesn't kind of exist before that point. So the RNA polymerase makes this, it pauses on the run of A's. And when this loop forms, right, there's a very limited space in RNA polymerase, right? There's just enough space for the two strands of DNA and one strand of RNA, right, to come through. And so this terminated loop actually forms within this channel, right, that's inside the RNA polymerase. And in doing so, it physically forces the RNA polymerase off of the DNA. And that's how it terminates transcription, right? So it's the formation of a structure within the RNA polymerase, right, that hairpin loop, that physically forces the RNA polymerase to dissociate. Now remember, it's not like a monolithic protein where we go like, ooh, if there's something inside of it. It's actually a complex of lots of different proteins held together in via a variety of interactions. And so essentially what happens is that complex gets pushed apart, and once it gets pushed apart, those interactions don't hold it together and just falls to pieces. That's intrinsic termination, right? It's part of the actual gene itself. Now we have what's called extrinsic termination or row-dependent termination. And it's actually funny here because I've, done a bit of reading on this to, understand it better myself. And it's actually a little bit, contentious, I guess, how exactly this works. So there's two models of thought. And kind of quite often what I do in teaching this is that, I'll teach you one of them, right? But it's worth bearing in mind, just the same as with, resolving recombination, holiday junctions and stuff, right? Remember I told you that this is one of the models, right? The strand invasion model. Well, this is one of the models of row-dependent termination. It's not the only one. And so it's actually kind of fun to, tell students that, well, we think it might be this, but it also might be this too, right? It's kind of hard to kind of resolve the differences. But essentially we have, again, a terminator sequence, right? So we have a string of A's, which slows down RNA polymerase. Again, this is another key feature. And we have a DNA sequence that encodes a bit of RNA that doesn't have any secondary structures, right? So we don't have anything that would form a structure that would get in the way of the Rho protein. So essentially what the Rho does is well, in this model anyway, it binds to that simple stretch of RNA with no secondary structures. And its role is to catch up to the paused RNA polymerase and boot it off the DNA, kick up the backside. And so that's why that terminator sequence that pauses RNA polymerase is so important, because if it isn't there, essentially Rho will never catch up with RNA polymerase and it will never terminate transcription. So that's why you got to have those two different things. So essentially what we have is, a simple bit of RNA, right? So no secondary structures, none of these loops or anything that would get in its way. And we have the RNA polymerase, which is busy happily transcribing DNA into RNA, right? So Rho binds and it's going to move along. It's like kind of like pulling itself along the RNA sequence as fast as it can. And RNA polymerase is moving along the DNA, making that RNA as fast as it can too, right? So then the RNA polymerase encounters that terminator sequence and it pauses, right? Kind of like just stays put, like, I'm not really sure what I'm doing, just stays there. And that allows Rho to come along and it basically kind of gets into that complex, right? So it forces off, this is called the torpedo model, right? It's like it's coming in and it just blows up the whole complex. It unwinds the DNA and the RNA complex, and it physically forces the RNA polymerase off of the DNA to terminate transcription. There's another model where it just kind of messes around with the RNA polymerase, but I don't understand that well enough to teach you. And I don't want to confuse you either. Right, so that's in bacteria. So bacteria, it's actually fairly straightforward, right? The termination is either built in to the gene itself through these inverted repeats and this terminator sequence here, or, I mean, again, essentially it's sort of built in because we have this unstructured RNA and the terminator sequence here, and we use Rho as a means to turn that into a termination event. Now, in eukaryotes, it's much less well understood, right? So we have one kind of known method. We just don't know if that is all of it, right? So this is the one that we do know. There are probably other means or mechanisms which we don't know of, right? Because we haven't found them yet. So one big difference here is that eukaryotes don't rely on terminator sequences or secondary structures in RNA, right? So that's a key part of intrinsic termination and Rho-dependent termination right, are these sequences, terminator sequences, the hairpin loops. Don't use that in eukaryotes. Instead, what we have is a cleavage site, right? So in eukaryotes do things very differently with regards to the mRNA, what happens to it, right? In prokaryotes, this mRNA, once it's done, well, actually, even as it's being produced, gets translated, right? So what you make from the gene, you get the mRNA, use that mRNA, you make protein. In eukaryotes, it's very different. So eukaryotes make a precursor, like a pre-mRNA. So this is an mRNA that is, it's mRNA, right? But it hasn't yet been finished, essentially, right? There's a lot of processing that takes place. So anyway, the RNA polymerase just keeps going on its kind of merry way, essentially, way past the end of the coding sequence. But in doing so, it basically transcribes what's called a cleavage site. So this is a short bit of DNA sequence that is recognized by an endonuclease, right? That comes in and it cuts it. So now, again, RNA polymerase is still, chuntering along, doing its own business. Now, our pre-mRNA is now free to go be processed, right? So that's off. That's going to go somewhere else. Oh, sorry, I got a question here. Well, a lot of it, Caitlin, is actually down to sequence recognition, right? So the whole assembly of RNA polymerase is guided by specific sequences in the promoter for both prokaryotes and eukaryotes. And this cleavage site is a specific sequence that's recognized by an enzyme. And so that enzyme will only ever bind to and cut DNA when it finds that specific sequence. So in a sense, right, the gene does encode its own sequence required for transcription termination. So in a sense, that's kind of similar to intrinsic termination. But no, well, really, there's a lot of stuff that goes on between cells that relies on different signals and the like chemical signals or protein signals. But within a cell, most of what controls gene expression is down to proteins that recognize specific bits of DNA, right? Specific DNA sequences, so or specific RNA sequences in this case, right? So they'll only do something if that sequence is there. If it's not there, it doesn't happen. Yeah. So that's the main driver. It's not all of everything, right? There's extra stuff that goes on, but most of it's based on the actual sequences of the DNA or the RNA itself. Yeah, it's kind of built in essentially. So anyway, once that RNA is cleaved, right, there are an awful lot of enzymes, RNases around, which will go, woohoo, free end, right? Yum. And they'll bind onto that free end and they'll chew it up. And so that's what RAT1 does, is it attaches to that free 5' end, right? RNA polymerase is still, off at the races, and it will chew it up. And essentially, this is like, I don't know if anyone's ever read the Langoliers by Stephen King, right? It's kind of a weird sci-fi novel. It's actually kind of cool. But they basically digest the previous, like the last day, right? So every day is a new day, and then the last one just kind of gets, degraded by these Langoliers. That's what I always think of. And so essentially its job is to chew up that free bit of unused RNA, which doesn't encode anything, until it gets to the RNA polymerase, and again, forces it off and terminates transcription. And that's that pretty much. So I'm a little bit over time, so I'm gonna love you and leave you. If you have any questions about this, have a look through this stuff again and then bring those questions with you on Wednesday. and I am more than happy to answer them. Cool. Have a lovely day, everybody. I'll see you all soon.